The story I'm going to tell you about today is about children that are born with a hole in their diaphragm. So every day there's about 150 children in the world that are born with this abnormality. They have a hole in their diaphragm and they struggle with breathing because their lungs are also abnormally developed. As a surgeon, I operate on these children. So on the right of the screen, you see a screenshot, a camera shot, when we're looking down from the chest towards the diaphragm. So this is the rim of the diaphragm. Oh, this is the rim of the diaphragm, and these are the abdominal organs that have herniated from the abdomen into the chest. So we operate on these babies after they are born, and then we close the diaphragm, and we make the hole closed with a few sutures. So in itself, that's not a very complicated surgery. However, the problem occurs after the surgery, when these babies are admitted to the intensive care unit, and that is not because of their abnormal diaphragm, that's because of their abnormal lungs. So the lungs in this disease are the main problem, and since the year 2000, over 300,000 babies have died from this disease. And that's mainly because of their abnormal lung development. So in our lab, we're trying to understand the lung development better, and also by doing that, then hopefully come up with a treatment to improve the lung development before these babies are even born. We use something called microRNAs for that. So everybody probably knows about DNA and genes. So if you consider a gene like a light switch that you can turn on and off, microRNAs are small gene regulators that function kind of like light dimmer switches. So they can regulate the gene expression. And we were the first, a few years ago, to discover that babies with this abnormality have different levels of a certain microRNA in their lungs. So when we investigated this in babies with this disease, we found that the babies that had higher levels of these microRNAs were having a better outcome. So this inspired us to try and find a way to use that as a prenatal treatment. So the lungs of these babies have lower levels of the abnormal lungs, have lower levels of these microRNAs. So we're thinking if we can increase the levels of these microRNAs, maybe the lungs are doing better. So we started off and actually brought a few people from my lab today who are doing the hard work while I'm doing other stuff. And they have figured out that if you use this in cells, so if you treat cells that are coming from lungs that are treated in a culture dish with these microRNAs, that they actually look better after the treatment. So in a culture dish, we're able to treat the disease. Then we went to lung explants. So what we did, we isolated the lungs, we put them in culture outside of the body, and then we treated these abnormal lungs with the microRNAs. And we were able, again, to improve the lung development in culture. The big experiment, of course, was to try and do this in an animal model. So we have an animal model where we use rats, and these rats have the disease. And then when we treat the rat pups when they're still inside the mother with these microRNAs, we found that they have less holes in their diaphragm, and they also have much improved lung development, to the point that normally they would die right after birth, but right now they would stay alive for the first few minutes while we were watching them. So that was a huge improvement from before, because we knew they would normally always die. So what I'm asking support for in this Innovators meeting is that we want to try and bring this treatment from the lab, where we have it right now, in animals, to the stage where we can start trying this in babies with this disease. Because we know that they'll have this disease around half the pregnancy, halfway during the pregnancy, because of their ultrasound. So when we know it now, we kind of know that we have to wait for a bad outcome. What we want to try and do is, when we diagnose it in utero, that we can treat these babies before they're even born and improve their outcome. Our ultimate goal is somebody like Kara. She's one of my patients, and she struggled a lot after birth when she was born with this, but we did the surgery and eventually was able to overcome all the difficulties, and now she's growing up almost like a healthy child. But Preferably, we would want to treat these children before they're born so they don't have to struggle as much after birth.
So this, in a picture or two, is what I study, and that is the early origins of health and disease. So over here we have a cover of Time magazine telling about how the first 9 months, so the time you're in utero, shapes the rest of your life. And then on the other side, the first 1,000 days. So this actually refers from the period from conception until 2 years of age. That's the first 1,000 days of our lives. And research is really showing that this is a critical period for development. So this is when in utero our immune system starts to develop, of course all of our organs and tissues, and then that early period after we're born, our brain continues to develop and our immune system continues to develop and our metabolism sort of gets set into a groove for life. So we think that studying this early period is therefore really important for understanding how our health shapes over the rest of our lives. So in particular, I'm interested in the early origins of these 3 chronic diseases. So asthma, allergies, and obesity. These are really the top 3 chronic diseases affecting children. They're major burdens on our healthcare system and our economy. So thinking of obesity, today 1 in 3 children in Canada are overweight or obese. And we know that this puts them at increased risk for obesity later in life, and that puts them at increased risk for things like cardiovascular disease and diabetes. Thinking of asthma, 1 in 6 Canadian children has asthma today. That's the most common reason for a child to come to the emergency room. It's also the most common reason for children to miss school. And if they're missing school, their parents are often missing work. So it's not only about kids being sick, it's about sort of the economic impact of these diseases. And then allergies. Hands up if you know a child or a person with a peanut allergy. So at least half the room. If I had asked a group of people that 20 years ago, probably no one would raise their hand, or maybe one person, right? So when I was a kid going to school, I had a peanut butter sandwich for lunch every day. That was no problem. Today, peanut butter outlawed in schools, right? And that's because so many people today have peanut allergies. And we don't really know why. Someone asked about genes in the previous talk. So we know that genes and genetics are important to health and diseases, and many of these conditions run in families. But thinking of something like a peanut allergy, this has skyrocketed in the last 20 years. And there's no way our genes as a human race have really changed over 20 years. So it's got to be something in our environment that has changed. We know that, but we don't really know what in our environment has changed and caused this epidemic of allergic disease. So I work with the Child Study, and we are studying 3,500 Canadian children and their families. We're studying the early origins of these chronic diseases with the goal of being able to predict who's going to get one of these conditions and who isn't, to treat those conditions better when they occur, and primarily, and that's why it's in the center, to prevent those disorders from happening in the first place. So I'm going to show you a quick video because I think this is the best way to introduce the study.
Imagine a world where allergies, asthma, and chronic illness are rare, treatable conditions. Better yet, imagine a world where these conditions can be prevented before they develop. 3,500 Canadian children and their families are helping to make that world a reality by taking part in a groundbreaking birth cohort study called the Canadian Healthy Infant Longitudinal Development, or CHILD Study. This is one of the largest studies in the world to look in depth at how genes and the environment interact to cause allergies, asthma, and other chronic diseases. Launched in 2008 with funding from Allergen, the Allergy, Genes, and Environment Network, and the Canadian Institutes of Health Research, the CHILD Study has recruited pregnant mothers and their families, carefully studying them until the children are at least 5 years old. Researchers are looking at the effects of outdoor air pollution, dust, chemicals, and pets inside the home, as well as moms' and babies' diets, stress, and antibiotic use. This involves testing drops of the baby's blood and taking samples from dirty diapers. Environment Canada is partnering with the study to better understand the impact of traffic pollution on the development of allergies and asthma, and Health Canada is testing urine samples to measure levels of phthalates, a harmful chemical found in soft plastic. Research is still underway, And by 2017, the child study will have collected more than 500,000 questionnaires and over 600,000 biological samples that will be available to scientists for decades. So, what has the child study discovered so far? Research is showing that pregnancy, birth, and the first 100 days of a baby's life are critical to health and well-being. By just 3 to 4 months of age, almost all Canadian children have been exposed to at least one phthalate. Babies living in areas with higher exposure to air pollution have higher risk of developing allergies, as detected by skin testing at 1 year of age. Babies born by cesarean section have lower levels of important gut microbes compared to babies born by regular delivery. However, at 12 months, this difference is reduced if babies are breastfed. What's more, breastfed babies have been shown to have a reduced risk of childhood obesity. Now that the oldest children in the child study are 5 and the youngest have celebrated their 2nd birthday, researchers are analyzing the data to find out what factors relate to health and disease outcomes. They are confident that their findings will influence public policy, parenting decisions, purchasing behaviors, and even urban planning, all designed to keep us healthier in a world where allergies and asthma become much less common. Of course, there is still much work to do. Today, 3 million Canadians suffer from asthma. 1 in 3 Canadians is directly affected by allergic disease, and 50% of all Canadian households are directly or indirectly affected by food allergies. These numbers are on the rise, and the child study with its 3,500 families will help us find out why, for this generation and the ones to follow.
So this is the child study, the Canadian Healthy Infant Longitudinal Development Study, or CHILD for short. Good studies have good acronyms. And so what we're really doing in this study, as I mentioned, is looking at the environment. Now, the environment is very broad. When I say that word, you might think of things like air pollution, and that's something we're looking at. But we're also looking at things like stress. Stress is part of our environment. Stress on children as they're developing, but also stress in mothers and parents when they're pregnant or in the early life of the child. We're also looking at lifestyle, so things like cigarette smoking, nutrition, so what moms are eating when they're pregnant, also what babies are eating when they're growing up. We're looking at infections in early life and how this tracks forward to disease later on. We're looking at socioeconomic status, so if a child is born into a family that's struggling versus a family that's better off, what impact does that have on their development? So we're doing this looking at the environment at a very broad level, starting from when moms are pregnant. So to really capture that first 1,000 days and beyond. The study is national, so we have 3,500 families participating across the country from 4 sites, so Vancouver, Edmonton, Winnipeg, and Toronto. We actually call ourselves the Manitoba site here because we're the only one of these 4 sites to include some families outside of the city. So we have some families participating from the Morden-Winkler area. And we think that's important to be able to look at exposures that are unique to rural families. So the big question is how genes and the environment interact to shape child health. So these are some numbers that I think are impressive and important about the study. We've had $30 million invested to date. It's expensive to do these large studies. By the end of this year, we will have collected 500,000 biological samples, so 500,000 samples. This includes blood to be able to do genetic analyses. It includes urine and stool. The stool samples from the baby's diapers are kind of my personal favorite. That's what allows us to study the microbiome, which are these trillions of bacteria living in us and on us, mainly in our intestines, helping to keep us healthy for the most part. We also collected breast milk. That's my new favorite sample. I'm doing a lot of research on breastfeeding and breast milk lately. So a huge resource for research. We'll also have had 200,000 questionnaires completed by these families by the end of this year. So we're connecting with them on a yearly basis. They're answering questions for us about their home environment, about their child's health. We're also seeing them in our clinic on a regular basis. The 3,600 families I mentioned, 93% is something I have to definitely highlight. So that's our retention rate. That's the proportion of families that we recruited when the moms were pregnant who are still involved in the study 5 years later. And that's really incredible. These type of longitudinal studies, it's really difficult to keep families engaged over that long period of time. So we are doing really well in this study at keeping our families engaged. There are many researchers involved. They cover many scientific disciplines. So you can imagine from the breadth of the environment that we want to capture, we need people who are experts in pollution, in microbiology, in nutrition, and these are all represented in our team. And then what I've circled on the side here is our timeline. So we're in 2017. By the end of this year, all the children in the study will have turned 5. And that was the original end of the study, but all of us researchers never really thought that would be the end. As you can imagine, after we've collected all this data and followed the families for all this time, we really want to keep going and see what happens next as the kids reach school age and beyond. So I'm going to highlight 2 studies, sort of 2 results from the study so far. These are 2 research projects that I was involved in. The first was looking at these gut bacteria that I mentioned. So we have trillions of bacteria living in our bodies. Mostly they keep us healthy. They help educate our immune system as we're developing. They help us metabolize foods. So through those functions, they can influence weight gain, allergic disease, asthma, even our mood. And so one of the first results out of the child study was looking at these gut bacteria and finding that they were different in babies who were born by cesarean section versus babies who were born normally in a vaginal delivery. And when you think of it, that's the first exposure a baby has to microbes in the outside world, is through the way it's born. And so we found these differences, and that was a really unique discovery, and we're following up on that with further research. The second result is about artificial sweeteners. So one of the mini questionnaires we collected in the study was from the moms during pregnancy. They filled out a questionnaire about everything they were eating. And one of the items they reported about was artificial sweeteners. And what we found was that the moms who were consuming these artificial sweeteners on a daily basis, compared to the moms who didn't consume them at all, their babies were at an increased risk of being overweight by 1 year of age. So a really interesting finding that had never been studied before in the world. We're the first to show it. And so we're going to the next step to now understand why that might be, and we have a variety of ideas that I'm happy to talk about at any point. But these are the types of interesting findings coming out of this large study, and these are just 2 examples of many. So I want to thank the people involved in this study. It takes a large team to follow all of these families, and we try to have a lot of fun doing it. So here we are having a Halloween party with the child study families. We make it fun for them so that they're coming back. We have 1,000 families participating here in Manitoba, and if you do the math, that's more than a quarter of the study, right? So we're hitting above our weight in Manitoba. I'm really proud of that fact. And so these are some of the people involved. It's my pleasure to work with them every day and do this research. And I'm hoping that through the Innovators program, we'll get some support to keep the study going and keep following these kids as they grow up.